All right, so this is going to be a very brief overview of the LKE model for those of you who haven't seen this. Okay, so starting with like a predator-prey model. So that you may have seen this before. This is like a log of Voltaire model with logistic growth. This is from the 20s originally, and then this logistic growth here is from the 60s put in. So this is kind of the classic predator-prey type model you might see in an undergrad ODE class when you start learning about predator-preys. You might see it in ecology class as well, rabbits and foxes, predators and prey. The point of this is that this is usually in terms of one currency, and usually we talk about things in terms of carbon. So I'm going to briefly just go through taking this model and turn it into a stoichiometric model where we're now going to add in phosphorus. And you can see some of the assumptions that I made there. Okay. So first of all, this is a differential equation. X is going to be the change in X over change in T is the change in the algae population over time. So this is tracking the carbon in the algae or the carbon in the prey. This is a differential equation for the carbon in the Daphnia or the carbon in the predator. So this is our X. We're going to take units for X to be carbons per liter. This is our Y. We're going to take the same units to be carbons per liter, but this is carbons in the Daphnia population. So we have two different equations here. One of them, again, is carbon in the algae, and one's in the Daphnia. And then here's some data under different levels of light for this model. So here I'm changing on the horizontal axis is just time in days, and on this axis is the densities. Sorry, this isn't data, this is model simulations. So you can see under some low level of light, I'm going to go back to the model real quickly, that's using to define the carrying capacity in the logistic growth expression. So under low levels of light or low inputs of carbon, you can see we get to what's called a steady state here. So you run that differential equation model in MATLAB or whatever, and you can see what's happening to the dashed curve is the algae population. So the carbon in the algae is leveling off, and the Daphnia population leveling off here. That's called the stable equilibrium. If we increase light, something interesting happens. All of a sudden, you see those populations are in flux, and those are the classic predator-prey type cycles you see in this predator-prey model. This is some data. This is actually the same data set that Hao showed you that Jutaro Urabe collected, where he did the same experiment, but he actually did it rather than just having MATLAB do it for you. So he manipulated different levels of light, kept track of carbon, kept track of the carbon in the algae and in the Daphnia population. So you can see under low light, algae is not doing so well because it needs that light. But the Daphnia population right here is actually doing quite well here. Under high light, you see that algae population taking up that level of light right here, so it starts to increase really fast and then drops off because of the predator-prey type cycle. So you see this high food quantity and you see this Daphnia population shooting up. This is almost looking like predator-prey type cycles. So these first two panels correspond nicely with our model before from the 1960s. Where this model breaks down is that when they put it under extra highlight, the algae soaks up the sun because it's doing this photosynthesis thing, becomes quite happy up here at these high levels. But despite this high food abundance, the Daphnia population is not able to thrive. This is because of the light nutrient hypothesis Hao was talking about. The third set of data here is the P to C ratio. So you can see under low light, they have this high food quality. Under high light, despite all this food abundance, this is low food quality. So this was that junk food that I was talking about. Okay, so this last panel caused them to think about, well, things aren't really just carbon. Let's throw something else in the mix. Let's throw phosphorus in the mix. And so that was the motivation for developing what's called the LKE model now. It was based off of this theory of ecological stoichiometry. L was low lead, so that's the L in it. K was Kwong, that's Yang Kwong right there who helped develop it. And E is Elser, this second author of this book here. So they took this model and they threw that phosphorus in there. What they updated was the carrying capacity. This is now the carrying capacity for the algae population. It's not just in terms of carbon, but it can be dependent on phosphorus as well. So they use Liebig's law of the minimum, which Hao explained earlier, to think about, this might be limited by phosphorus or carbon. They modified this with that minimum law with a minimum function. Right? So they put in carbon and phosphorus. Now algae can either be limited by carbon or limited by phosphorus. They did the same thing with the Daphnia growth rate. The Daphnia could be limited by carbon, but it could also be limited by phosphorus now. Use the same law of the minimum, let's modify this. So they modified this to include a minimum function, which now takes two inputs. One is the original model, so it's Daphnia limited by carbon. The other one is now a new model, which the minimum would take this Q over theta, which is now Daphnia limited by phosphorus. Okay? So here's that full model, and then they have an expression for Q, which I don't have here. I should have put on there. But so Q is going to be a function of x and y as well. Okay. So this. Yeah. Yeah, that's a good point. Okay. So then taking this basic model where they put in Liebig's law of the minimum to make the carrying capacity or the algae growth dependent on carbon or phosphorus with a minimum threshold approach. Same thing for the Daphnia. These are the new simulations here. So you can see under very high light, you get this, what they were hoping to get, where you have high light, you have the algae population doing well. Despite that high food abundance, you have the Daphnia population dying out. So comparing that with the data is promising. And again, that's what we call the LKE model. So a lot of the models that we're talking about, or that I work on, are based off of this. And so the model that Howe showed at the very end there, which was a toxicant, the paper at the end, which was a toxicant nutrient model, is basically taking Howe's toxicant model and coupling it with this stoichiometric model here and kind of pushing those together. So hopefully that's helped. If anyone had any questions, hopefully that was helpful. Any questions on that?
One comment I can add is because of the experiment by Chitala, they also sort of accidentally introduced some competing species. So that data sort of suggests that at low quality, there's actually they can high quality so that's sort of like this is a simple model setting. You can also sort of like expand that. So in a way that sort of the single model can be useful to not just expand this data set, also the one that which I think it's in the paper.
Yeah, in the Jafaro paper. Okay. So I'll skip some of that. Some of the analysis for now. We can talk about that in our small groups. And like I was saying, we took the LKE model, we threw a toxicant in there. So this was the model that Howe showed you, a predator-prey toxicant model. These highlighted functions in red are toxicant, so like a toxicant growth, but it doesn't depend on phosphorus. So we just modified these with some assumptions. One is that we're going to use a stoichiometric approach where you see the minimum function here, but you also see these maximum functions, which is the toxicant approach, or the toxicant dynamics. So we're assuming a linear dose response here for both the predator and the prey's growth. And so we modified that model by just kind of coupling those two, and mushing those two models together, taking the LKE model, which has these minimum functions, and taking the toxicant model, which has the maximum functions here. And we made this toxicant-mediated predator-prey model. This is what we've been working on, and it's in JTB from 2016. But we made a lot of assumptions when we made this. It might not be accurate. It's the linear dose response thing is something I wanted to be, have this group kind of discuss in this breakout session about to happen, whether or not that was an appropriate assumption to make. And this model was motivated by that methyl mercury data that Roxanne was talking about earlier. And we used that to parameterize it. Yeah. Yeah.
Thank you. So I understand, can in the simple model, can it handle luxury uptake of phosphorus by the algae? Because what I wonder is what some of the discussions that have come around is the intersection of these potentially toxic compounds are going to occur with nutrient-rich conditions, and so this model sort of stresses the effect that occurs due to potential phosphorus limitation of Daphnia, but what about pushing it in the other direction? Okay.
Yeah, this was the LKE model where they looked at phosphorus limitation and how it affects grazers' growth. My dissertation was actually on the other side, where if you have too much phosphorus, how does that affect growth? So we have updated this model to incorporate excess phosphorus and how that hinders growth of Daphnia outside of toxicology. that hasn't been looked at in terms of toxicology yet. But that could be something to work on. But we certainly have, this model can certainly handle looking at,
Yeah, I mean, given Paul's data, I think that's actually the more correct way to go at this point, right? Because we're talking about growth rates. Well, whether I don't know what happened with your model. I need to read your paper. But if growth rates continue to increase as food quality increases, or if there's some trade-off to that,
so
then either way, it changes the result, the toxin interaction.
Yeah, it does. So what they've seemed to find is that growth rate does increase with food quality, right? So as you increase phosphorus, you increase growth rate. But what's being called the stoichiometric knife edge is actually if you get too much phosphorus, you actually start to decrease that growth, what they're seeing. And one of the arguments or hypotheses why that occurs is actually if you have too much P in your food, so too much phosphorus rich, you're not eating enough. So you actually have a reduced feeding rate because you're following some simple rule where you're going to eat enough phosphorus until you get enough phosphorus, and then you're good. And so you're actually starving yourself of carbon. So there's these experiments where they have Daphnia that's surrounded by lots and lots of food, lots and lots of algae that has a really high P ratio, and they're just starving because they're not eating enough, even though there's plenty of food around them. So that's something that they've seen empirically, and that's what my model showed that I worked on. Coupling that with toxicants, that's why we're here to figure that out. Yeah, good point there.